Today we're going to extend our conversation about macronutrients and biological macromolecules by running chemical tests on food items. In this experiment, we are going to be using what are called colorimetric tests to measure with color. Here's the idea. We have a chemical indicator, and this indicator is built to detect one type of molecule and has a starting color, right? It has color 1. If you add it to a substance and the target molecule that you're looking for example, if there's protein in it, and this is an indicator that detects protein, plus target molecule, then what'll happen is you will get a color change, a particular kind of color change. So we have different types of indicators for different classes of food molecules, of biomolecules. So if I have a mystery food item, and I want to know what's inside it, there could be all kinds of things inside it. There could be protein, which I have an indicator for. There could be carbohydrates, which I have 2 different indicators for. There could be lipids, which I have an indicator for. There could be other things, but those are going to be what we test for today. And I add, let's say, my protein indicator to that mystery food, and I get no color change. Now this would imply that there's not a lot of protein in that food item. There's at least not enough to trigger the chemical reaction that gives us a color change, because if there was, then this would have changed colors, this little protein indicator chemical that I added. But you can't just rely on the results of this test. You need a control for comparison. And here's why. What if this indicator is expired? What if it's gone past its shelf life and it's busted? Maybe it's expired. Maybe it's the wrong indicator. Maybe it got mislabeled, something like that. You can't just assume that the experimental setup is correct. So what you need is what's called a positive control. A positive control is a test that, if everything's going correctly, should give you a positive result. So in this case, instead of my mystery food, I'm gonna add egg whites to my protein indicator. And then what does that do for me? Since I know that egg whites contain protein, that's basically all egg whites are, is protein, and I have this protein indicator, I am expecting a color change. If I do not get a color change, that means that the experiment is broken. If I don't get a color change, I can't rely on the result up here, because now I know that it won't even change color if there is protein present. So a positive control is necessary to validate some of the results of your experiment. It shows you that you're not getting a false negative in this case. Whereas you also need a negative control for the opposite reason, to prevent you from getting false positives. So if I take water, distilled water, something that cannot possibly have any organic molecules in it, if all of my labware is clean and I went through the process of getting the distilled water properly, it can only have H2O, no protein, no sugars, nothing else inside. It's just water, no organic molecules. And I add that protein indicator again, I should get no color change, because there's no protein present. If I do get a color change, that means that I have some kind of contamination, or there's something wrong with my indicator, or something else has gone wrong with my experimental setup. So you need positive controls and negative controls in order to validate any kind of results you get from your experiments. So that's what we'll be using in this experimental setup. We're going to begin by testing our indicators using positive and negative controls, and then we'll move on to testing some food items. The chemical indicators we're going to use today are iodine, which can be used to detect complex carbohydrates like starch or cellulose, but it can't detect simple sugars like glucose and fructose. Sudan is used to detect lipids like vegetable oil over here, so it'll detect oils. We have burette solution, which starts out as blue but turns purple in the presence of protein, so we're going to use egg albumin or egg whites as our positive control for that. And then we have Benedict solution, which changes color in the presence of reducing sugars like glucose and fructose, so simple sugars. We're going to start out by running the positive and negative control tests for the burette solution, which again detects protein. Our negative control is regular old water, and then our positive control is going to be the egg whites. So you can see I'm filling these two test tubes up. One contains water, one contains egg whites, and to each tube I'm going to add the burette solution. Now burette starts out with this kind of light blue color, as you can see here. But in the presence of protein, it becomes this lovely lavender, violet, purple kind of color. So the result of this test should show you what a negative result and a positive result look like. Here we'll shake it up. The water tube looks like it's just light blue, and I wanted to make sure it showed up on the camera. This is a qualitative test, not a quantitative test, so I didn't feel necessary to keep track too much of how much it is, but here you can see the purple coloration coming in there. Now we can see it on camera much easier. So purple for burette means protein, blue means no protein. Next we're going to test out the lipids, so I'm going to fill one tube up with pure water, and I'm going to fill another tube with vegetable oil, with soybean oil. And I'm going to use Sudan 4 as my indicator for lipids. Now this one doesn't actually change color. Sudan preferentially clings to lipids, so it'll spread around in regular old water like any kind of dye. It'll just kind of mix up and become a dilute light pink substance inside that water. But in the presence of lipids, it will cling to those oils, so you'll see it stain the droplets of oil rather than the water. So here you can see, I mix it up, but because water and oil remain separate, the Sudan stays clinging to that top layer of oil. Next test, we're going to test out the iodine solution. So I'm going to add some cornstarch to the water, mix it up to make a little cornstarch milk. Now cornstarch is a complex polysaccharide. It's a complex carbohydrate, lots and lots of sugar molecules, glucose molecules linked together. And in the presence of complex carbohydrates, iodine, which starts out as a kind of dark amber color, a kind of light brown color, will turn a bluish black, a purple blue kind of black. So you can see the amber color peeking up there. And here I used a little bit too much iodine. I got overzealous. So what I ended up doing was diluting it in the tubes so that you could see the color difference here. So I'm going to add some water. And as it gets up here, you can see the light shining through, and you can see that this tube on the left, which did not contain starch, preserves that dark brown amber sort of color. But the tube that did have starch actually turned black. Iodine turns black in the presence of starch. And if I take a sample of this dark water and I put it into this cup and mix it around, you can see it's not just black, it actually has a little bit of a bluish purple hue to it. You can see it just making it through in the light. It's not a dramatic color of blue-purple, but you can see just a little bit of a difference between that amber, regular old iodine and the dark blue color that it changes in the presence of starch or other carbohydrates. Last, we have to check Benedict's solution. So once again, our negative control is going to be water, regular old water. There's no sugars in there. Benedict's solution looks for what are called reducing sugars, glucose and fructose, monosaccharides. And our positive control is going to be a glucose solution. So I'm going to bring in a heaping spoonful of glucose powder, stir it up, and then we're going to add this glucose solution to the second tube. Now, if you look at both tubes, they look pretty much the same. They look like they both contain water. I'm going to label this one W and this one G for water and glucose so I don't get them switched. Now I'm going to add the Benedicts. One eyedropper full into the water, one eyedropper full into the glucose. If you take a look here, they both look the same still. They both look that light blue, and that's because Benedict's needs an extra element. Benedict's requires heat in order to get its color change. So I'm bringing in some hot water. I'm going to place the tubes into the hot water, and I've time-lapsed this, so it's going to happen a little bit faster than it normally would. With a little bit of sugar, Benedict's will turn green. With a little bit more, it'll turn yellow. With a little bit more, it turns orange. With a little bit more, it turns red. So you can actually get an idea of how much sugar is inside your food based on how much color change you see with Benedict's. And now we can see the difference. The negative control, water, no color change whatsoever, but the positive control, the glucose solution, turns this nice deep orangey red color. That's a positive result for Benedict's. So now that we've seen what the positive and negative results for each of these indicators look like, let's test some foods. I'm going to squeeze some orange juice here, make some fresh squeezed. And of course it got really sticky on my gloves, so I changed out my gloves. And I added a little bit of water so that we could see the color change. We only needed a dilute mixture. Added an equal amount of orange juice to each one of these tubes, and then to each tube I'm going to add a different indicator, starting out with the Sudan on the far left, which detects lipids, then the iodine, which detects complex carbohydrates, then the burette, which detects proteins, then the Benedict's, which detects reducing sugars. I'm going to put the caps on because I'm going to mix these up. And of course, Benedict's requires heat. Now you can see that it actually starts out green here, but that's because the Benedict starts blue and the orange juice has the kind of yellow color. So of course you're going to add blue and yellow and it gets green. But as we add heat, it shifts over to being a much darker orange color. So that is a positive result. It went from blue and orange makes green to orange and orange, giving us a really deep orange color. So we got a color change, and that's positive for reducing sugar. So it makes sense because there would be fructose inside fruit. Meanwhile, the other results don't have any positive indication there. They all are negative results. No color changes there. Next food item. How about some spaghetti? I'm gonna crush these up. Make some spaghetti in my mug using my tea kettle. Took a while for the spaghetti to boil down, but once it was ready, I extracted the water from around it, and I put an equal amount of water into each one of these 4 tubes. And then, just like before, I added the indicators. First, the Sudan, then the iodine, then the burette, then the Benedict's, and of course, adding the caps, mixing them up. We'll have to put the Benedict's on heat in a moment. No result for protein in the spaghetti. But look at that! Look at the black, the bluish black coloration that we got here. That is a positive result for starch, for carbohydrates, complex carbs in spaghetti. And no lipids to speak of. That looks exactly like our negative control did, like our just pure water did there for the Sudan 4. So that is a negative result for lipids, but we have a positive result for complex carbohydrates. What about reducing sugars, simple sugars? Look at that. Went from that nice light blue base color that Benedict's has, and now it's obtaining a kind of cheery yellow, a canary yellow coloration. So that is a positive result for monosaccharides, for reducing sugars in there. So we have 2 positive results for spaghetti. Next up, garbanzo beans. They are pre-cooked before put in the can, and then they are placed in with some of the water that they were cooked in, so all I have to do is actually extract the aquafaba from around the beans and place it into the tubes, and then I'll use the rest of this can for dinner later. Same order as before. I'm going to use the Sudan first, iodine, burette, which I can see is turning purple immediately upon going in there, but it wasn't showing up very well on camera, so I do add a little bit more burette just so the camera can pick up the coloration. That is a black color change for the iodine solution. That is positive for complex carbohydrates in the aquafaba, the bean solution. You can see a little bit of the purple color, but then when I shake it up, it kind of dissipates. You can see it a little, but I decided to add some more burette. Once again, this is not a quantitative experiment where the exact amounts are being measured out with delicate care. I just want to show positive and negative results. So as long as it shows up on camera, that's my goal for today. There you go, you can see the purple coloration a lot better now. Now the Benedict's, of course, you know how this works by now. It's got this kind of darkish blue color, but we need to add heat. This footage is sped up about 20 times faster than reality, and in that you can see that there is a subtle color change that does occur. It goes from a kind of murky blue color to a slightly more pure brown kind of color. It's definitely the smallest color change that we've seen with the Benedict's, but there is a very small color change. So there are some sugars, some monosaccharides floating around in this bean juice as well, just not as much as we saw in some of the other food substances. One thing I want to note before I close out this video is that pretty much all foods are going to contain all kinds of macromolecules. Since all of our food is made from living things, and all living things are made of cells, and all cells have lipids and proteins and nucleic acids and carbohydrates, pretty much all food is going to contain all of those macromolecules. The question is just in what proportion, in what amount. What these tests show you is what macromolecules each food is particularly rich in, which ones it is a very good source of. So we've learned that orange juice contains monosaccharides, spaghetti contains monosaccharides and polysaccharides, garbanzo beans contained a little bit of monosaccharides, but lots of polysaccharides and lots of protein as well. The other main thing that I hope you took away from this video is the difference between a positive control and a negative control, and how you can use them in order to validate the results of your experiment.